Morning.
Good morning.
Good morning.
Morning. First, any questions or concerns?
I have a couple questions, but I'll wait until the end of the class.
Okay. All right. Are they questions with regards to content, Zena? Are there questions about material? Zena, are they questions about material or are they questions about the class?
Well, it's just about like the quiz that I was supposed to do. Like,
okay, all right. Okay. Yeah, if it was questions about concepts and so forth, I was just going to ask you to ask them, because a lot of times if you have them, other people have them.
That's true.
And they may not even realize they have them.
That's true.
You know what, Jade, if we hear Elmo in the background, we'll be all right with that.
I am so sorry. I just figured I would at least let you guys know. I'm like I'm going to unmute myself, and if you're happy and you know it, clap your hands in the background. So
we like Elmo. We can do Elmo. So no questions. Let's get started. One thing, I'm going to mention this now, and then I'll mention it again when we start lab. I am going to ask that you send me your home addresses. And the reason is because I'm going to mail you some urinalysis strips. And to do that, I need home addresses. And I'll tell you that what happens is I fill out the envelopes, I send them in the mail, and then I shred your addresses. I don't hold on to them. But I want to make the lab, even though it's remote, I like to make it as hands-on as possible, which, you know, is not a lot. But this is one of those labs that we can make hands-on. And it's a urinalysis lab. And so I have boxes of strips here. And I just want to send you some in the mail. And so don't give them to me right here. And what I'd prefer, you can either text them to me or email them to me, but just send them to me. And then I collect them. And once I have everyone who wants to be involved with this, I send them out and then I shred your addresses. So I'm going to ask for your addresses. And let's get started. So we're starting into the immune system today. And we spent some time talking about the immune system last week in lab. But we'll really delve into it today. I told you about the book that I read with regards to the immune system, this Immune by Philip Dittmer. And I highly recommend it. I listened to a lot of books. And I don't recommend every book that I read. I don't recommend a lot of the books that I read, especially to students. But this one is really, really good because the immune system is fairly complicated. If you think about all the parts that we were talking about last week, it's fairly complicated. Nevertheless, it's exceptionally powerful. And this book describes it on a layman's perspective and puts it into a story. So I highly recommend it to you. He's an author of a YouTube channel that explains everything from science to technology to whatever. I honestly have never subscribed to the YouTube channels, but I really did enjoy that book. So if there's things, I'll quote some of the things I heard from the book and we'll go forward. Because I thought, you know, I wish I could teach like that sometimes. So bear with me. So the lymphatic and the immune system, chapter 21. As we talked about last week, the lymphatic system is working in collaboration with the circulatory system or the cardiovascular system, in that tissue is being leaked out from the capillaries. Sorry, not tissue. Fluid is being leaked out from the capillaries. And it goes out and circulates around the cells. And that's what we call the interstitial fluid at that point. Is circulating around the cells. And then, because we only have 5 liters of blood, and part of it's now circulating around in the cells, it has to be returned to the blood. And so the lymphatic capillaries, that's one of their major functions, is to return that, all that, some of that fluid, a major part of that fluid. And I think your book gives a number. And I think it said maybe around 17% or 13% or 20%, somewhere in there. And so it picks up this fluid in these one-ended tubes or closed-ended tubes. So not like your vessels where they've got an open and closed end to them. Sorry, not like your vessels where there's 2 open ends. I'm apologizing already. Give me 1 second to get my mouth in order. Okay. So they're closed end tubes, so they're only open on one end. And so it pulls up that fluid. The fluid actually, it drains into them. And they're taken up by the lymphatic capillaries. When the lymphatic capillaries are going to merge and become collecting vessels. And collecting vessels are going to become lymphatic trunks. And lymphatic trunks are going to become collecting ducts that are going to drain into the 2 subclavian veins. On the right side, that subclavian vein is the right lymphatic duct. If we wanted to, we could call the thoracic duct, which is on the left side, the left lymphatic duct. So major functions of the lymphatic system. It's involved in filtering the lymph. You take her out or you go back to bed? Taking my teaching assistant. So filtration of the lymph, because there's a lot of things that can be taken up in that interstitial fluid that are circulating around the cells. Lots of things such as pathogens, wastes, ions, solutes, all sorts of different things. And they return that lymph to the blood. Also, the other function of the lymphatic system is that it's involved in the production and training of the immune system lymphocytes. And one other additional function that the lymphatic system has is it's involved in absorbing and transporting lipids in the small intestines via the lacteals, which are specialized lymphatics that are specifically involved in absorbing lipids and lipid-soluble vitamins. So lymphatic capillaries surround the blood capillaries. The lymphatic capillaries become lymphatic vessels, which in conjunction with lymph nodes and tonsils and the spleen are involved in filtering that lymph. The lymph nodes and the vessels that are leaving the lymph nodes, the efferent vessels, are going to merge to become lymphatic trunks, which then merge to become lymphatic ducts, which then drain back into the subclavian veins. So lymph vessels, one-way vessels that surround capillary beds with valves. And we'll talk about the fact later today and right now that because these lymphatic capillaries, just like veins, are under such low pressure, they have to have valves in them to prevent the backflow of lymph. Keep it moving one direction. So the only thing that's really pulling the fluid into the lymphatic system is the increased pressure in the interstitial fluid, in the interstitial tissues or the surrounding tissues. The increased pressure pushes on the overlapping cells, allowing for the fluid to move in. Questions so far? Thoughts? Thinking about the lymph node and the lymphatic system as a whole, we have tonsils, we have lymph nodes, we have lymphatic capillaries, we have lymphatic vessels, we have lymphatic trunks, and we have lymphatic ducts. Remember that we have additional tissues such as the Peyer's patches, the aggregated lymphatic mucosal malt associated lymphatic tissue. Another thing that we call these Peyer's patches are malt, which, bear with me here because I don't think I have it on this PowerPoint. We often think of the Peyer's patches as being these, which are malt. Mucosa associated lymphatic tissues. These would also include the mucosa and mucus that's throughout your trachea and within your nose. Areas where we can, entrances where we can where incoming pathogens can be trapped. And they're trapped, and at the same time, they're exposed to cells of the immune system. Guys are really quiet this morning. So if you have questions, let me know. There was something else I should say about this. Remember the drainage patterns. The drainage for the thoracic duct is the whole, the lower legs, the abdomen, and the left side of the trunk, arm, and head. The right lymphatic duct is going to drain the right arm and the right side of the head and the right trunk, or the right chest area. What forces drive this? We haven't talked about the circulatory system, but it's the same forces that drive the blood that's moving against gravity, that's going to move from your legs towards your heart. So if you think this is fluid moving in a hose, just like blood is moving through hoses or vessels. Hoses works better because a lot of us are familiar with garden hoses. It's got the same what's the word for it? Same mechanisms of flow. So different forces that drive it are the skeletal muscles as they contract and relax. They're pushing the fluid forward. Another one is that the respiratory pump. As you breathe in and out, it creates a suction on your lower body, pulling the fluid up towards the heart. Another one is just that these are closed end tubes. and therefore it's accumulating at one end, and there's no backflow. There's no backflow because there's valves. And therefore, as it accumulates in the closed end part of the tube, it's going to, as just a concentration, it's going to be driven towards where it's a lower pressure of fluid. Are there questions about the drivings? Let me actually do this. So driving forces. Lymphatic fluid as it's returned to the circulatory system. So lymphatic flow. Skeletal muscle contraction. Second one is the respiratory pump. And let me actually back up to the skeletal muscle contraction, because this is one of the major ones when it comes to moving the lymph from your feet back towards your chest. And I want you to keep in mind here that these are the same types of forces, at least these first two, that are involved in moving the blood from your feet towards your chest and your heart.
I'm sorry, did you say the first two? The driving forces of the lymph flow, and then
no, these two, the skeletal muscle contraction. Let me give them numbers. One. So the driving forces are the skeletal muscle contraction and the respiratory pump. We're going to talk about a third one, but these first two are also driving forces in moving the blood from your feet back towards your heart. So the same forces that are involved in moving blood that's under low pressure back to your heart are involved in moving the lymph back towards your heart, and therefore draining it back into the circulatory system next to your heart in the subclavian veins. Does that make sense? Is that clear? Somebody tell me yes or no.
Yes, thank you.
Okay. The last one that helps drive this fluid back is that these are closed-end tubes. All of your lymphatic capillaries are closed end on one end. Therefore, as the fluid is going to accumulate at one end, because all fluid moves from a low pressure to a high, from a high pressure to a low pressure. That means that as it accumulates at one end, it's going to build up a pressure there. And because there's also valves in here to prevent backflow, we'll talk more about valves this afternoon, but recognize that these valves are there to prevent the fluid from moving, forcing it to move in only one direction. Questions?
Mm-mm.
So blank are the largest of the lymphatic vessels, and they empty into the what?
Lymphatic trunks.
A.
A.
I was going to say C.
I would say, let me ask you a question. Let me say this a little differently. What do we call those parts of the lymphatic system that drain into the subclavian veins? What are they called? What are they called? Somebody go back and look in a few slides. Tell me what they're called. What are the vessels that empty into the subclavian vein? Somebody go back and look.
Is it D?
Why? Well, Lauren, why is it D?
I'm not sure. I just remember you saying that the collecting ducts emptied into the subclavian veins.
Oh, you're right. You're exactly right. So if we come back and we look about right here, so the lymphatic ducts are what are going to return to the venous system. Another way, looking right here, actually not right there. Did I not have a good picture of it? Right lymphatic duct that's draining into the subclavian vein. The thoracic duct, which is also a collecting duct, remember we could also call it the left lymphatic duct, is also draining into the subclavian vein. So the answer, like Lauren said, it is D. D is in Delta. I don't know if I mentioned this because I can never remember what I mentioned in some classes and what I haven't mentioned in others sometimes. But looking at these questions that I'm posing here, when you've taken the quizzes, have you seen them there? If you've taken the first quiz, did you see any of the questions that I bring up in the PowerPoint?
I don't recall seeing any of the questions that you posted off.
Okay. Well, I only post a few of them in the PowerPoint, but let me tell you, these are the same, coming from the same pool of questions that I'm using on your exams and quizzes. And so what I'm trying to do with these is get you thinking in the right direction, because really the whole purpose of this class is to prepare you for exams and about ways that your knowledge is being evaluated. And so know that all these questions are also, they're coming from that same bank of questions. So now, specifically looking more at the different organs and tissues. Primary organs, the thymus, which is the site for T cell development, and the bone marrow, which is the site for B cell development. Other organs of the lymphatic and immune system, the skin, the lymph nodes, the malt. Remember, this is mucosa associated lymphatic tissue. Let me scroll in just a little bit more. Mucosa associated lymphatic tissue, which, you know, I said it was found in the respiratory system. I said it was found in the small intestines. It's also found in the gastrointestinal system, because all these areas where bacteria and pathogens could possibly enter your body are going to be coated with this mucosa associated lymphatic tissue. In the small intestines, the reason it's there, do you remember from last week in lab why we have it in the small intestines? All of the food has already gone through the stomach. Why do, and that the pH in there is so low and so acidic that we tend to destroy all things. So why do we have it in the small intestines? Why are these Peyer's patches so critical? Where else do you possibly have a lot of bacteria sometimes? Oh, really? Just so you guys bear with me. We have a student who can't get in, and she was just told to call the welcome center for some reason. And now the welcome center isn't letting her into the class either. So the welcome center isn't even open today. So give me 1 second here, bear with me. And I guess all of a sudden she's been dropped from all her classes for some reason. And I don't even know who it is. So give me a second. All I have is a phone number here. But let's see here. Yeah, I know it's somebody we know. Bear with me. Who is it? It's Casey. And I don't know why she's been dropped from all her classes. Look.
Could it be a financial issue? Because when I started classes, like, I was really confused. Yeah. And I didn't know how to pay for the classes because, like, I literally just graduated and no one told me how to do adult things. So I didn't know how to pay for my classes, and they said that your classes would be dropped if you didn't pay. So maybe that's what she's going through.
Hi. If I give you, I can't give you access to D2L. But if I give you the meeting ID number, do you think you can get into Zoom?
Yeah, I should be able to get in.
Let me give you the ID number. Give me 1 second here.
Okay.
Because it's great when they tell you to call the welcome center, and the welcome center isn't even open today. One second here.
Okay.
Cancel. I'm a little brain dead as I was last week. I'm not as brain dead as I was last week, but I'm still a little brain dead. So give me 1 second to get my act together here.
That's okay. Take your time.
I wrote down the Zoom meeting number.
All right, Becky, what's the Zoom meeting number?
I have it as
Casey,
9.
Wait. Do you have your pen and paper, Casey?
Yes, I'm ready.
Okay, go ahead, Becky.
All right, it's 9, 6, 8, 3, 4, 7, 7, 2, 8, 9, 1.
9, 6, 8, 3, 4, 7, 7, 2, 8, 9, 1.
Yep.
Okay, we'll try.
So Casey, stay on the phone while we do this, because the other thing is you could just listen in via our phones.
Okay, yeah. Okay.
There's a couple different options here. See if you come up. Yeah, I was wondering where you were at.
Yeah, I've been trying for a while. Okay. I think it's one of my
Even though we're an online class, I tend to get used to who's in this class. And because the other reason that I tend to get used to who you are in this class, every one of you, is because you tend to be vocal. And so I know who you are, which is great. It's great.
Okay,
it makes me feel like I'm not listening to talking to myself.
Right.
So let's see here. Is it waiting for the host? Is that what it says?
Yes.
Well, you're not coming. Where are you? Have you logged into the myhack.edu first?
Yes.
Let's see here. Let me see if there's another way to do something here. Okay, what number did she give you again? What ID number?
Excuse me. 968.
That's not it. That's not it.
Oh.
Let's try this one. Let's try this one. I'm not sure if it's right or not, Becky, but let's try this one.
Okay.
989 9640 0969.
Okay.
Try that one now. The other thing is I can email you the link, but let's try this first. There she is. Awesome. Okay. All right. Okay, I'm hanging up. Awesome. Okay. And you students, I put my phone on vibrate because if I didn't, it would disrupt me the whole entire class. But if that happens to you and for some reason you can't get into the class, start trying to get a hold of me. And I do keep my phone next to me, even though I put it on vibrate. So I'm looking down at it occasionally. So just let me know when you can't get in and I will work to get you in. I'll try. I'll do what I can to try to get you into the class every time.
Thank you so much.
You're welcome. All right, let's come back into this. Or were we?
You were doing the primary organs.
Okay. And why do you think we have that mucosa-associated lymphatic tissue or the Peyer's patches in the small intestines?
Bacteria.
That are coming from where?
Oh.
Because they're not coming from the stomach, because the stomach is already really, really acidic and it kills almost everything.
See.
What's another area in your body outside of your esophagus, your respiratory system, your urogenital area? Where else is it possible that you could have a lot of bacteria?
Would it be like your liver or your spleen?
Not really, because they're pretty closed off. Where else?
Mhm.
Let me be, and you'll find you kind of know this already. I kind of get grossed out by a lot of things, like as Becky's there hacking out a lung. But have you ever had to do a poop smear at the hospital? Some of you.
I've had to collect it. Yeah.
Why do they do that?
Cologuard.
What was that?
I've had to do Cologuard, so
Oh, say, what is that?
that's when you poop in a bucket and send it in for cancer screening.
Oh,
You do your own little smear.
all right, that's just as nasty.
They do it to test to see if they're what bacteria, like if somebody has diarrhea, sometimes they do it to see if there's C. diff or what's going on with it.
Okay, so on that idea, why do you think you have Peyer's patches in the small intestines? You guys, you're right. You're looking right at it. Why, if we have it in the large intestines, do we want it? We don't want those bacteria going other places, right?
Right.
We need them to stay in there.
So it would be the small intestine.
Right, exactly. They're essentially right where they are is right. If I remember right, I don't know if they're throughout or if they're in one specific place, but I think they're generally in one specific place, which is right next. Bear with me here. I'll show you a picture and that will help. Scratches. So Peyer's patches are concentrated. I don't know how well you can read that, so let me read it to you. Peyer's patches are concentrated in the distal 25 centimeters. And if it helps you to keep this in mind, your small intestines is approximately 21 feet. So 25 centimeters of ileum, which is the last part, which is the most distal, closest part of your small intestines to your large intestines, but extend approximately 200 centimeters. So what they are is they're there in case there's any escaping backflow of bacteria from the large intestines. Because you've heard of people getting their bowels perforated on accident during a surgery and so forth, and that they worry about sepsis.
Yep.
They're worrying about it for this reason. We really like the bacteria in our small intestines, our large intestines, because it does a lot of things for us. But we also need it to stay there and not go anywhere else. If you read the book that I was recommending to you, he talks about some pretty grody examples of why that bacteria is so important. I'm not going to go into it with you because it'll just make me kind of sick. It kind of made me sick in the book. But some of you are pretty used to dealing with poop coming from the large intestine. So you might understand that even better than I do. He made the comment that we like it because it actually isn't, those bacteria are involved in synthesizing vitamins. They're involved in breaking down some foods even further. There's a lot of different reasons to have them. but we need them to stay where they're at. So in lymph nodes, tonsils, the spleen, the let's see here, lymphatic vessels, these are all secondary organs, lymphatic organs. Primary lymphatic organs are the thymus and the bone marrow.
You said the secondary, I'm sorry, was what? The tonsils, the spleen?
Everything here, except for the thymus and the bone marrow. So the lymph nodes, the tonsils, the thymus, the spleen, the mucosa-associated lymphoid tissue or MALT that we find in the respiratory tract, in the gastrointestinal tract, in the genitourinary tract. And part of the gastrointestinal tract is the small intestines, or AKA that MALT within the small intestines being the Peyer's patches. So moving on here just a little bit. This is just kind of a summary slide. Primary organs, the bone marrow and the thymus gland. The bone marrow in adults is diffusely located in central portions of flat bones, such as the ribs, the sternum, the pelvis, and the vertebrae, with smaller amounts found in the epiphyses of some of the larger long bones, such as the femur. The major function of the bone marrow is to act as a major hematopoietic tissue, producing all the formed elements of the blood, including the erythrocytes, the red blood cells. You remember urethra means red. So cytes is cell, so red blood cells. Leukocytes, leuko means white or clear, so white blood cells, and the platelets or thrombocytes, because remember we know thrombus is a blood clot. Sometimes if it helps you to remember what other names platelets can be called, because that reminds you what they're involved in, what their function is. Thymus gland is a small butterfly-shaped gland located in on the superior surface of the heart. It has a major role when you're a child and a developing embryo / developing baby. And then it starts to atrophy as you grow up. And its major job is to train the T cells. So it plays a key role in programming the immune system to recognize self and provides a site for the maturation of T lymphocytes. We'll talk more about this in a second, but I told you about the book that I really liked. That book talks about the thymus as being the university of death, where if you can't pass, you die. Oh, yeah. He says he talks about how awful school is for college students. And then he says, but if you think you've seen hard classes, imagine dying because you can't pass. Okay. The spleen is the largest lymphatic organ. It has a lobular structure and it's located on the left-hand side of the abdomen between the 9th and 11th ribs. It helps trap foreign material and remove damaged and aged erythrocytes, while also acting as a reservoir for erythrocytes, leukocytes, and platelets. The spleen is where erythrocytes also go to die when they're too old. Lymph nodes are the small bean-shaped lymphoid organs found in the major clusters, including the cervical, axial, and inguinal lymph nodes. And they primarily filter lymph, trapping the foreign materials such as bacteria and viral particles. Questions about these organs specifically right now? So major barriers to defenses. One thing that I hope you gain from this is how powerful your immune system is and what an amazing system it is. So you have three major barriers to defense against invasion. You have non-specific physical and chemical defenses. Your skin, told you about this book that I love, talks about the skin as being a hellhole where it's acidic and there's nothing on it except for a bunch of bacteria that protect your skin. And therefore, unless you can penetrate it, you're probably not going to survive on it as a bacteria. The mucous membranes, there's a bunch of slime in there, and pathogens and viruses and all sorts of unwanted things get stuck in that stuff. And on top of that, there's a bunch of macrophages in there that go through and eat everything that gets stuck in it. It's kind of like a spider web where things get stuck in it and then the spider comes along and eats you. Stomach acid. Very, very low pH, it just tends to destroy things because it's got a pH of 1 or 2, about the same as hydrochloric acid. Lysozymes. What are lysozymes? Do you remember that from a really long time ago when we were talking about in chapter 3 in 121? You remember that?
Isn't that something that's like found in like tears and saliva, if I'm not mistaken?
Tears and saliva. That's another thing that we don't mention here, but saliva is another one of these non-specific physical defenses because of the lysozymes. And what does it do in there, Jade? Do you know?
Doesn't it lead to like cell death or something?
It does. It does.
Oh, okay.
Think of lyso as lysis. And so these are enzymes that lyse things. And so you've got tears in your saliva. You've got lysozymes in your tears. You've got lysozymes in your saliva. And they just destroy foreign stuff. They destroy things that shouldn't be there. They destroy intact cells. Expulsion is why you have coughing, sneezing, vomiting, and diarrhea. Just to get rid of that stuff once it gets broken down. Innate immunity or natural immunity, remember this is a non-specific immunity, is the second layer of barrier, which includes macrophages, neutrophils, eosinophils, basophils, and monocytes. Remember, monocytes actually become macrophages. So maybe not the best way to replicate that and make you think that they're two different things because they're not. One more, the complement system. Another one that's here that we don't see, natural killer cells. Do you remember those from last week? They're the ones that go around and they don't, when they see something odd, and what they look is if there's the absence of something. When they see something odd, rather than try to ask that cell if they're doing well, if there's something wrong, can they help them? They just put a bag over their head and shoot them. Because that's their job, is if it looks odd, just kill it. Adaptive immunity is a cellular or humoral adaptive immunity, or acquired meaning specific. So these are the three very major lines of defense. Starts with non-specific physical and chemical defenses, non-specific innate immunity, and then adaptive acquired specific immunity. So innate or non-specific immune system. This is the defense system with which you're born. It protects you against all antigens. It's also allergies. Allergies are also part of the adaptive immune system, but we'll talk about that later. The innate immunity involves barriers that keep harmful things from entering your body, your skin. This is the first two lines here. And these barriers are going to form the first line of defense. The adaptive immune system, the first and second line of defense. Put that in there too. The adaptive immune system is receptor mediated. And think every time you see the word receptor, what that means is that it's specific. Something specifically has to bind to that receptor. If you remember when we last semester, when we talked a little bit about the endocrine system and we talked a little bit about neurotransmitters, if there's not a receptor for it on the cell, they don't respond to something. Unless a receptor is there for something to bind to, it's not going to respond. So think of this as being the specific adaptive immune system, where with the assistance of helper T cells, B cells will differentiate into plasma cells that then can produce antibodies against specific antigens. This humoral immune system deals with freely circulating antigens from pathogens or outside infected cells. Another one that's not included here that I should include here is cell-mediated immunity. And that's specifically the T cells. Humoral immune system the humoral immune system is specifically talking about antibodies and the B cells and antibodies. Actually, let me do this. The cell or cellular mediated immune system is specifically looking at T cells and their interactions with cells. And let's say direct interactions or or cell to cell, and their cell, let's say this, their cell to cell direct interactions. This is going to be crucial later. So two different types of the adaptive immune system. Humoral immune system, which involves B cells and their antibodies. And the cell-mediated immune system, which is T cells and their interactions, which involve cell-to-cell direct interactions. Or cell-to-cell, let's say rather cell-to-cell contact interactions. So the innate immune system is the first line of defense. It's non-specific, and its responses work immediately when the body is exposed to a threatening agent. It non-selectively defends against foreign invaders. It doesn't matter what the foreign invader looks like. If it's foreign, it goes after it. It's rapid. It happens immediately, but it's also limited in its responses because it isn't specific. Neutrophils, macrophages, and several plasma proteins are important to this type of defense. So the innate immune system is always on guard. It's a rapid response. And it's limited but non-selective against every and all invaders. Its major job and its major point of reason for existing is to contain and limit spreads of infection. If you can't tell, I really like the immune system. It's a powerful. This book kind of opened my eyes to a lot of things, but if you want to realize how powerful it is, think of autoimmunity. You know people who have struggled with autoimmune disorders. When it goes awry. So different structures and defense mechanisms of non-specific innate immunity. The skin is a physical barrier to penetration by pathogens. Its secretions contain lysozymes. This is the one that Jade was telling us about, an enzyme that destroys bacteria. So we see lysozyme in the skin, we see it in tears, and we see it in saliva. Let me write that down. Lysozyme. Found on the skin. Bear with me here as I figure out how to type. On the skin. Tears. And saliva. All of these places where bacteria can easily come in contact or enter the body. Another, and I don't think this is mentioned in this table, another one is the normal flora on your skin, in your respiratory, down your trachea, in your urogenital tract, in your large intestines. Told you we have to be careful with those large intestines, but they are part of the immune system because of their existence, they stop other things, other bacteria from taking up residence there. So normal bacteria or flora. I think I might have mentioned this. I used to, you can tell I'm an animal person. I really like animals. And for a very, very long time, I was involved with the humane system. Since I've moved to the East Coast, I haven't been involved, but I think at some point in my life, I need to get back into it. But I used to foster kittens, and I learned that one of the fun things about, not so fun things about having little baby kittens is that they get ringworm really, really easily. And ringworm is awful if you've ever dealt with it. It's a fungal infection. It sounds like a parasite, but it's actually a fungal infection. And I had a kitten for a while, and we finally figured out what was wrong with her. But when kittens lose their mommy, sometimes us people are really poor mother cats because we don't have all the tools we need to take care of them. And so she got ringworm, and I took her into the vet because I couldn't figure out why she, first off, she was just showing all the signs of being failure to thrive. And I took her into the vet and found out that she actually had a UTI, and that was part of her problem. And the vet said, I know she has ringworm, but if we don't start working on this UTI, because the longer UTIs go on, the harder they are to treat. She said, if we don't start going after this UTI, it's only going to get worse, and she's just going to get dehydrated and possibly go into kidney failure. And she said, so I want to put her on an antibiotic, but it's going to cause her ringworm to go insane. And I said, well, put her on the antibiotic. Ringworm is not lethal. UTIs can be lethal if they're not treated. And so she put her on the antibiotic. And why do you think that caused the ringworm to ravage her body?
Because the antibiotic is essentially like immune suppressant drug, so it makes it easier for the ringworm to spread.
Why would it be immunosuppressant? What would an antibiotic do, Martin?
Basically just flushes your body of good or bad bacteria.
Yeah, it gets rid of all the bacteria. And so you have the reason you don't generate regular fungal infections on your skin is because you have bacteria on your skin that keep that stuff in check and keep other bacteria from being able to take up residence and other fungi from being able to take up residence. And so when we put her on that antibiotic, it started working on the UTI, but it also killed a lot of the bacteria on her skin, which was working on killing the fungus. And so it wreaked havoc for a little while longer. So coming back to this, in addition to lots of lysozyme, we also have regular bacteria. So
sorry, just I don't know, curious, but how did they get rid of the fungus then? The worm.
I learned a trick. If you ever deal with ringworm, hit me up and I will save you from it. But she gave me a hint. She gave me a really neat thing, and I've actually had vets tell me that it doesn't work to do this. But I have what's called it's a veterinarian hospital sterilization fluid. And they use it to sterilize instruments in veterinarian clinics. My mom wonders if they actually used it when she was a practicing diabetes nurse practitioner to sterilize her equipment, even though it says it's only for veterinarian hospitals. But it actually will kill the fungus within a matter of days.
Wow.
And it was pretty amazing, because as I'm watching her, and I wish I'd taken a picture of it, but I realized I would have been teaching A&P a couple of years later, I would have taken a picture of it. But I realized, even if she's losing all the hair on her limbs, because I'm also treating it with this substance called Nalvazon. I'll put it in here what it's called. And don't tell your vet, because I had a vet actually tell me to my face that it would not kill ringworm. And I was not very impressed at that moment, to say the least. Bear with me here. But it's called Nalvazon. And what it does is it's a non-toxic, doesn't hurt to go on the skin. It's not like rubbing alcohol or hydrogen peroxide, in that it hurts to go on the skin, and maybe if you ingest too much, you can get sick. The animals can lick it right off their skin, and it's not going to hurt them. And you put that on it, and within a matter of days, it goes away.
Wow.
And it's amazing stuff. I actually have some in my back room that's concentrated. Best $36 I ever spent. And
I just looked it up, and it says that you can use it after an injury, before surgery, or before an injection. So that's super cool.
It's amazing stuff, and it doesn't hurt. Like when you put it on an injury, it doesn't sting like rubbing alcohol does. It's not like hydrogen peroxide, which kills good and bad tissue. And therefore, if you don't want scarring of a cut, don't put hydrogen peroxide on it, because it just makes it worse a lot of times. I had a cat, one more story. I had a cat who got a bungee cord hook caught in his mouth. And you've seen what they look like, little hooks that look like this. And so I came home from work one day, and he'd probably been struggling with that thing for hours and hours. And it was a Saturday night. It was actually a Valentine's Day a couple of years ago. And he'd probably been going after that thing, trying to get that thing out of his mouth for hours, because his face was all swollen, his eye was disoriented. And not knowing what to do, and probably doing the right thing, I got the hook out of his mouth. Well, his face was still horribly disoriented, and he had a lot of blood in his mouth. And there's a reason I'm not a nurse or a veterinarian, because I don't really have the stomach for that kind of stuff. But I got it out of his mouth, and then, between my friend and I, we got him over to the veterinarian clinic, and it was a Saturday night. And so the veterinarian was the ER veterinarian, the veterinarian on call. And she came in, and she was able to clean him up the best she could and send him home. And when I got home, I realized that I had a major gash, and I kind of have a scar from it. I don't know if I can even see it anymore. I had a major scar, a gash that went all the way down my arm, where when I had gotten that hook out of his mouth, he had gashed me with his claw because it probably really hurt. But so I got home and I look at this cut and it's easily 2 or 3 inches long. And there's just dried blood all the way down it. And I didn't even realize he had done it because my adrenaline was, I had so much adrenaline at that moment thinking he was gonna die on me, that I didn't even notice he'd gouged me so good. And I went home and I put some of that Nalvazon stuff on it. And if you've ever seen cat scratch fever, it's a real thing where you get inflamed from cat scratches. It's a reason that cat scratches and cat bites can be worse than dog bites. Well, I woke up the next morning and it had healed up and it didn't even get inflamed. And I probably left it unattended for probably close to 3 or 4 hours. And so all I did was go home and swab it down with this Nalvazon. And the next morning it wasn't even inflamed and it was healing up. And amazing stuff. So if you end up with cats with ringworm, I had a student in my anatomy class at Stevenson who got a kitten with ringworm last semester. And I gave her a little bit in a bottle and she said it was gone within a week. But I don't know why I went off on that. Bear with me here. Let me come back. If you ever need it, I highly recommend it to you because my mom and I have had the same bottle since we moved in here. We don't use it very often, but it's great when you need it. Okay, coming back. Normal flora. So you have bacteria in your large intestines, your skin, your stomach, your, not so much your stomach, but your esophagus, your trachea, your mouth, lots of bacteria in your mouth, lots of bacteria in your urogenital tract, lots of bacteria in your large intestines that are all there in their own way. They do specific functions, but a lot of them are there to keep other bacteria and other things from taking up residence there. So the respiratory tract, secretion of mucus, movement of mucus by the cilia and alveolar macrophages. You have specific macrophages in your lungs that work to get rid of pathogens there. The genital urinary tract or the urogenital tract, the acidity of the urine, the vaginal lactic acid, the sperm that enter the vagina are actually attacked by the immune system up to a certain point. Internal phagocytic cells ingest and destroy bacteria, cellular debris, denatured proteins, and toxins. Interferons, they're great. Think interferons, they interfere. They interfere with the replication of viruses. Complement proteins promote the destruction of bacteria and enhance the inflammatory response. And endogenous pyrogen. Remember what these are from last week. What's a pyromaniac? Guys, someone tell me what a pyromaniac is.
Someone who likes to start fires.
Yeah.
Someone who likes to start fires. So pyrogens, their major job is to induce heat and trick your hypothalamus into raising your body temperature. Natural killer cells. These are the cells that don't ask any questions, don't try to ask if your cell is all right, they just kill it. So they destroy infected cells infected with viruses, tumor cells, mismatched transplanted tissue cells, mast cells. They release histamine and other mediators of inflammation. They're similar in a lot of ways to basophils, one of the white blood cells. And their cytokines promote adaptive immunity. So here's another diagram showing the different barriers of the innate immunity. Lysozymes, the normal flora, skin, your stomach and small intestines that have rapid pH changes, the mucosa, the malt that we don't mention here, mucosa associated lymphatic tissue, your blood proteins, your white blood cells like neutrophils and basophils and eosinophils that are all part of the innate immune system. When we're thinking about the skin, the pores of the sweat ducts put out a bunch of lysozymes that is secreted onto the surface of the skin. Excuse me, and prevents things from being able to take up residence on the skin. Questions? So the different phagocytic cells and their locations, neutrophils, found in the blood and all tissues. Monocytes, remember these become macrophages, found in the blood. Remember these two are considered two of the 5 white blood cells, types of white blood cells. Remember that never let monkeys eat bananas. So never let monkeys, never neutrophils, monkeys, monocytes. Tissue macrophages, all found in, or histiocytes, found in all tissues. We have some dendritic cells that we talked about, the dendrites last semester when we were talking about the neurons. Dendritic cells are not part of the nervous system. So don't confuse them with dendrites. But what they are is they are macrophages. They phagocytize things. Kupffer cells in the liver. Alveolar macrophages in the lungs. One of the neuroglia, remember there were a lot of different types of neuroglia from last semester, but one of them was the microglia, which are the macrophages of the central nervous system. Questions? So phagocytosis, by definition, what is phagocytosis?
It eats bacteria.
It eats bacteria. It eats bacteria. It eats
foreign.
Yeah, it eats
foreign anything.
Foreign anything, dead anything. I'm realizing even if we made it through the immune system all the way today, I'm going to add a little bit to the end because there's a few things that I left off here. But pus is essentially a bunch of dead macrophages and neutrophils that have engulfed all the dead stuff and all the pathogenic stuff they possibly can. And then they just die. But phagocytosis is cell eating. And the first step of phagocytosis is what we call chemotaxis and adherence of a microbe. Because I know that Zena's the crazy one here. She's the one taking 121 and 122 at the same time. Looking at this picture right here, Zena.
Taking 2 classes, 2 biologies.
Yeah, she's taking 121 and 122 at the same time. Oh. I know, kind of nuts, huh, Casey?
That's ballsy.
That's a lot of work.
Nobody else would ever do that. Looking at this, Zena, what kind of microscopy was used here to get this picture of a macrophage?
Right.
What kind of microscopy was used here?
I don't know, honestly.
Wait,
the microscope.
What?
Like the microscope? Like
yeah. What kind of microscopy, Claire?
It would have to be like a computerized. I don't know, one of the ones that are more recently created.
It's definitely more recent.
I forget the names of them, but they're like computerized and right.
Let me give you a hint. There's light microscopy, which we're usually the most familiar with. And then there's transmission electron microscopy. And there's one more. What's the third one, Zena?
I don't remember.
Zena, go back and look. Google it. What's the third one? Google it. The nice thing about an online class and technology, just Google it. What is the third one called? Claire didn't have 121 lab with me. Scanning. So this is scanning electron microscopy. And the reason you know that, and the reason we know that right off the bat is because we're looking at the surface of the structures. We can see their surfaces. So what this is this is a macrophage that's adhering to a bunch of bacteria. And it's putting out these little pseudopodia to bring them in also, the bacteria in also. So the first step is chemotaxis, meaning the macrophage has to move towards the bacteria. Taxis meaning to move, chemo meaning towards a chemical signal. And then adherence to that bacteria, we have to be, the macrophage has to be able to bind to it. So the first thing that happens here is we get phagocytosis, which is, it moves towards the bacteria, it puts out these little pseudopodia. What does pseudo mean?
False.
Yeah, false. It looks like it, but it's really not. Podia, what's a podiatrist?
Foot doctor.
A foot doctor. So pseudopodia are false feet. And so the cell puts out these little pseudopodia the macrophage puts out these little pseudopodia and essentially brings the bacteria in and kind of brings it in and eats it. And so it brings it in, and then once the antigen is brought in, it's a phagosome, called the phagosome. And the phagosome in this vesicle is going to fuse with a lysosome. A lysosome is one of those organelles that contains a lot of lysozymes, like Jade was saying, that break things up. So the lysosome fuses with the phagosome, and then it's called a phagolysosome at that point. So the enzyme and the antigens mix, and the antigen is degraded. Bear with me if you're not sure what an antigen is, because what it is just surface markers on the bacteria. And we'll talk about it again here in a few minutes. But it takes in that antigen, mixes it up, breaks it all up because of the lysozymes, or the enzymes that are in the lysosome that break it all up. And then a lot of the antigens is expelled from the cell via exocytosis. Some of it is actually processed and attached to these MHC molecules, or major histocompatibility complexes. And it's displayed on the plasma membrane, on the external side of the plasma membrane. And I told you I really like this book. So if I'm going to quote things from this book, because I thought, I would love to be able to teach the immune system the way he did. What these cells essentially do is they bring in the pathogens, they bring in all this foreign stuff, they break them all down, and then they attach it to carriers and stick it on the outside of their body. All the foreign stuff, they break it all down, and then they attach it to the MHC molecules, and then they display it on the outside of their bodies. And so if you want to think of it in a really grody way, you eat something, go and you decide you're going to be a little carnivorous. You eat something, and then you spread its guts all over your body so that everybody can see what you've just eaten. That's what this is. Keep that in the back of your mind for right now, because it's going to be really important later. Does that make sense why phagocytosis is important here for right now? Is that it's saying this, guess what we found in the body that we got rid of? And if it's here, guess what? There's more of them. Just like if you see one cockroach in your house, know that there's 100 more. Shifting gears a little bit, interferons. Cytokines that are released non-specifically from any cell infected by a virus. Think of a cytokine as being a hormone that's specifically secreted by a cell. Any cell. Think cyto, and then kine is something that it's just a protein signal that it secretes. So it's released non-specifically from any cell, interferons, released from any cell infected by a virus. Provides a general and rapid defense until more specific but slower responding immune systems can begin. So these interferons, the different things that they do, They transiently inhibit the multiplication of viruses in most cells. They trigger the production of virus-blocking enzymes in potential host cells. They enhance macrophage phagocytic activities. So they enhance phagocytosis of other macrophages, stimulating, which ultimately is going, because of these cells are smearing all this, the guts of what they just ate all over their bodies and saying, look what else, look what's in the body right now. This stimulates the production of antibodies against those substances and boosts the power of natural killer cells and killer cytotoxic T cells. It also has anti-cancer effects. It also upregulates antigen presentation to T lymphocytes that are, remember, lympho lymphatic, sites meaning cell. So these are T lymphocytes are going to become T cells, and increases uninfected cells' abilities to resist future infection. So these are secreted by viral infected cells, and these are the different things that they can do, the interferons. So they stimulate macrophage phagocytosis. I think repetition is good here because we've got so many players. We're looking at a game here, a puzzle, and we've got a lot of different players. So interferons that are secreted from viral infected cells stimulate macrophage phagocytosis, upregulate the activity of cytotoxic killer T cells, upregulate the activity of natural killer cells, and increase the production of antibodies.
I have a question.
No, see if I can answer.
What's the difference between the three of them then, lysosome, macrophage, and phagocytes, or do they just all do the same thing?
Macrophages are one form of phagocytes.
Okay.
Natural killer cells don't phagocyte things, they just kill them. What was the rest of your question? Because I'm sorry, I am brain dead this morning. That's why I keep going on bad tangents.
The lysosome, like what's the, I was saying, what
lysosomes,
between the three
lysosomes are organelles within the phagocytes that their job is to destroy what the phagocytes bring into them. Zena, the benefit of you taking this class is you're helping these students who have had 121 get a really good review. So lysosomes are organelles within phagocytes and within all cells outside of red blood cells that work to destroy what's been brought into the cell or incoming things. It's they're kind of the stomach of a cell in that they break things down that are brought in.
Okay, thank you.
Because phagocytes, their major job is to break things and break up things. They have lots of lysosomes. And natural killer cells and cytotoxic killer T cells, they just don't, their job is not to break things up and display them later for other cells. Their job is just to kill things. So cell division. So interferons inhibit cell division. They inhibit virally infected cells from being able to divide. They inhibit tumor growth. They also inhibit the maturation of adipose tissues, so maturation of fat cells. And they inhibit the maturation of erythrocytes. How do interferons prevent viral replication? So a virus enters a cell. It replicates within the invaded cell. The cell then releases interferon because the infected cell then releases interferons. This is a way of a cell, once it's infected with a virus, is doomed to die. There's no way out of that one. But it kind of its last stand against the virus that's now infected it is to release this interferon. And then we talk about what are the benefits of the interferon. But this is kind of a viral infected cell's last stand before it dies anyway. So virus enters a cell, it starts to replicate in that invaded cell, and the invaded cell then releases interferon. The interferon binds with the receptors on uninvaded cells, so neighboring cells that are not infected. The uninvaded cells produce inactive enzymes that are capable of breaking down the viral messenger RNA and inhibiting protein synthesis within themselves. So interferons get taken up by uninvaded cells, and those uninvaded cells, as a result of taking up that interferon, produce enzymes that at that moment are not active, but if necessary, will be able to break down the messenger RNA if a virus tries to infect that cell. If that uninvaded cell that's now made these inactive enzymes that can break down viral mRNA, if they are infected by a virus, the virus blocking enzymes that this uninvaded cell previously made are now activated. And the virus is unable to multiply in the newly invaded cells. You guys, I'm not going to repeat that, but I think this is a good moment to take a break just for a second. Let's take 5 minutes or so. Actually, let's make it 7 minutes. Get up and move around, and we'll come back to right here. We love that stuff, right? mind. And it actually has more. I'm realizing some of the quotes just how much I remember. Because he talks in that book about how the interferons are essentially a virus infected cells last stand before it dies. Last stand before it dies. You know, once it's infected, there's no way out of that. But it produces these interferons that then go out and work to protect other cells against infection. I was looking through that book really quickly last night. Actually, pretty amazing book. Well, you knew that, right, Mom? Oh, cool. That's what. Something I listened to about when he was a young father and his sons. I love that. Yeah, it was women's. It was women's conference. Oh, is that what it was? With his spirit? Yeah. I think that, you know, that's an amazing. Yes, that is an amazing story. You know, it's one of those things where they can break us with it so quickly. Be careful what you do. Before it goes stopping from doing something very much. Oh, I don't know what you shared that with. So take that way. Wow. Yeah, it was a women's conference a couple years ago because I was living. I think his son was jumping on the bed or something. Just grab it like that. Since I very carefully put that down. Well, you know, if it had been really, I think if it had been, I think a lot of times spirit, you know, it's when it feels also getting it has to. But it also will intervene when it's very important. You know, depending on the relative, you know, because you don't know what under this year was essentially telling that was a no-go. Well, he was tuned in, but you know what? You don't always have to be. I don't think you always have to be. I think it comes differently depending on the situation. I remember yesterday this class like yeah. I'll bet you Henry Byron had those kind of things too. It just doesn't mention. Does that make sense? Doesn't mention that kind of stuff. Going to school yesterday and trying to figure out how I was going to put the class together. I'll have some time to I'm making it. So I'm driving in my car and it's a quiet moment. This is why we do work in this car. Because I'm just not very good. My head is tends to be very in a lot of different directions all at the same time, which is good because it allows me to do what I'm doing right now. But at the same time, it means that sometimes it's really hard for me to organize things into something logical. Apologize. All right, come on. Class. Okay. Sorry, I don't know how much time that was when I'm back. So blank are secreted by cells infected with viruses, alerting neighboring cells and protecting them from becoming infected.
B.
Say it one more time, Jade.
B as
B
in brat.
Yep, B is in brat. Yes.
Sorry, my kids are home, so I'm just like my daughter.
I wasn't even going to mention that. I thought because she's there having to listen to Elmo all day.
Yeah.
But we won't say anything. You gotta, Jay realized they grow up. You gotta love them while they're this little.
I know.
So another part of the innate immune system, the complement cascade. I don't know if you guys heard me as I was talking with my mom. I realized as I stood up, and I'm always so brain dead on Fridays. And I told her just as I was walking into the other room, I said, I love this class. So thank you guys for making this a good experience. Several proteins in the complement cascade additionally augment the inflammatory process by serving as chemotaxins. Do you remember what I said chemotaxins were from earlier? What are chemotaxins? What are chemotaxins? Chemotaxins, taxon is essentially to move towards something. Or taxis, it's also taxon or taxus, meaning to move towards something. And so chemotaxins are essentially signal chemicals. They work like sirens, saying this is where the infection is at. Come this direction, because this, you cut your foot, and within a very short time, you have a bunch of chemotaxins there sending out siren signals to other cells, telling them this is where the infection's at. They also act as opsonins or opsonins. What this means, opsonins means to make more delicious. Opsonins make pathogens look yummier for other white blood cells to eat. They make them more likely to be destroyed and eaten. They also promote vasodilation and increased vascular permeability. So vasodilation meaning the increasing the diameter of blood vessels, and increased vascular permeability meaning things are leaking out of the vessels, increasing the amount of leakage out of a vessel into the interstitial tissues. They stimulate release of histamine from mast cells. Histamine is another siren, bringing in more bringing in more white blood cells and fluid into that area, and activating kinins, which are more signals. Or more chemo, or more inflammation mediators. So specifically, how does this work? How does the complement pathway work? Well, it gets activated through 3 different mechanisms. The first one is the classical method, or the classical pathway, which is where an antigen binds an antibody, and then the complement proteins see it, and it initiates the reaction cascade of the complement proteins. The alternative pathway is antibody dependent, so we don't need an antibody. Or antibody independent, sorry. Classical pathway is antibody dependent. The alternative pathway is antibody independent, where complement, all these C3, C4, C5, whatever, the C always means complement. So C3 dissociates into C3A and C3B, and C3B binds to a pathogen surface, cell surface. This is the alternative pathway. And when C3B binds to that pathogen surface, it initiates the reaction cascade. The third way is that a protein called lectin binds to the carbohydrates on a pathogen's cell surface and signals activation of the complement cascade. So 3 different ways. The antigen-antibody complex forms on the pathogen surface and stimulates the complement reaction cascade. The alternative pathway is that C3 itself directly binds to the pathogen surface rather than an antibody. And the third way is that lectin, a protein, binds to carbohydrates on the pathogen surface instead of C3 or antibodies, and it initiates the complement cascade. So at the beginning of this activation of these reaction cascades, C3 dissociates into C3A and C3B. C3A will combine to basophils. Remember what basophils are. They're one of the white blood cells. So C3A combine to basophils and mast cells, which are similar to basophils in that they also release histamine. Therefore, when C3A binds to basophils and mast cells, it initiates the release of histamine and other inflammatory promoting chemicals. And the result is that we get lots of inflammation. C3A also stimulates neutrophil and macrophage activity. Remember, these are phagocytes. Neutrophils and macrophages are phagocytic antigen-presenting cells. And so this will also increase inflammation. C3b, when it associates from C3a, can bind to antigen-antibody complexes on red blood cells. And red blood cells then can transport those antigen antibody complexes to the liver and the spleen, where phagocytes in the liver and the spleen are going to remove and degrade those antigen antibody complexes, resulting in immunoclearance, just getting rid of those complexes. C3b can also coat bacteria, viruses, and other pathogens remember, as an opsonin. That's why it coats them, making them look more delicious to phagocytes, resulting in them getting eaten and destroyed. The last way that C3b, of the three ways that C3b works, is C3b can split and activate C5. And C5 splits into C5a and C5b. C5b will then bind C6, C7, and C8. Remember what that C means. These are just different complement proteins. And this C5b, 6, 7, 8 complex binds to a ring of C9 molecules, forming a membrane attack complex, or MAC, that then penetrates the pathogen surface, or viral infected or cancer infected cells, not cancer infected, but cells that are undergoing cancerous processes. Then this complex perforates those cells and lyses them. So right here, it's showing just that it inserts into the cell, into the plasma membrane, and lyses them. This whole thing is the complement pathway. Any questions? So MAC, pretty amazing little destructive structure, forms a pore-like channel in the plasma membrane of targeted cells, resulting in leakage from those cells and ultimately cytolysis and destruction. Questions? So the C3b protein coats proteins. So, because I can't read all of a sudden, the C3b protein coats bacteria and stimulates phagocytosis by what? During a process called what? So what cells are involved in phagocytosis? Looking at the list of cells here, what cells are possibly involved in phagocytosis?
B, the neutrophils and macrophils.
E, right. And what is this process by which the protein coats the bacteria? What is that process called?
opsonization, however you want to say it.
However you want to say it, opsonization. Yes. Opsonization. So remember, opsonization means to make delicious or make more tempting. So they're just it's coating it and making it more, making the neutrophils and macrophages think more about destroying it. Kind of like adding seasoning to a soup just to make it a little bit more tasty. Complement fixation cannot lead to what? What do you say? Go back to this slide right here. Ultimately, this is what's going to occur by the complement cascade. So complement fixation, or think of fixation as being activation, cannot lead to what?
C.
Say it one more time, Jake.
C as in cat.
C as in cat. Endogenous, right. Endogenous pyrexia, what is that? That's the pyrogens causing increased temperature. So innate non-specific immunity, exogenous pyrogens or endogenous cytokines are released by leukocytes that are going to trick the hypothalamus into thinking that we need to be at a higher temperature. And the hypothalamus in the brain is going to reset the body temperature. What outside of making you miserable, like my mother's been running a low-grade fever since like last Saturday or last Friday. Outside of a fever, what that makes you miserable, what are the benefits of it? It can denature toxins. Remember, you have enzymes have optimal temps where they function best. Bacteria have optimal temps where they function best. And heat is one of those ways that we can denature a protein. Remember how you can fry an egg, which means that you denature the proteins. So a fever can denature the proteins and slow the growth of bacteria or kill it. Also, it causes you as an individual to slow down so the body can repair itself. A pyrogen is a substance that causes what?
E.
Say it one more time.
Oh wait, no, sorry. Is it A?
It's not. What else could it be? What's a pyrogen?
E.
Would it be E?
It's E as in echo. Because remember that a pyrogen, think of a pyrogen as being a pyromaniac. It's going to cause increased heat. What causes increased heat of the body is going to be a fever.
Mm.
And you guys keep in mind this complement fixation, AKA this is also complement activation. So moving on a little bit further, innate, nonspecific, natural killer cells. These are naturally occurring lymphocyte-like cells. They non-specifically destroy virus-infected cells and cancer cells. Their mode of action is to directly lyse cell membranes upon first exposure to these cells. I think I've mentioned this in the past, so you know that I like to read, and I like to read books of all kinds. And sometimes I like to read very graphically violent to a certain level. And one of my favorite shows is NCIS. And so I love watching it. And one of my favorite set of movies is the Bourne Identity series, the Bourne series. And so when I was reading this book by the Immune, and one thing that he says about these natural killer cells is they don't ask any questions. You know, they see a cell and they recognize that there's something not quite right about that cell. And mainly it looks for the absence of what is normal. And if what's normal is not there, rather than asking the cell how it can help, it puts a bag over its head and shoots it.
It's like a hitman.
It's a hitman. And he doesn't ask any questions. He just kills it.
He's got a job to do and he's doing it.
Exactly. So it's the mode of action is to directly lyse the cell upon first exposure and kill it. So these natural killer cells are non-specific. They're just looking for anything that isn't normal. And they're providing a temporary fix until cytotoxic T cells can be primed and mature. Remember, these are non-specific, so these are the first line of defense. The cytotoxic T cells take time to adapt and get ready and be primed. And so all of these natural killer cells and phagocytes and complement, they are just holding the line until the adaptive immune system is where it needs to be to take care of things and get rid of it ultimately and be done. And, oh shoot, why did I think I messed this up, but you know what? I think I switched some slides around, so bear with me here. We'll come back to those natural killer cells. Inflammation. Actually, give me one second. I don't like, for some reason I did things a little wrong here, I'm realizing. Where did I put them? I know they're here. Bear with me. Okay, you guys, we're going to talk about this and then we're going to go back. So bear with me here for a second. I should have put this slide up with those other slides. And I will adjust it in between lab and lecture and lab. So the natural killer cell goes up and if you, the way that it's depicted in this book is it essentially goes up to the cell and licks its face. You can imagine having, some of you have kids and you've gotten that wet and slobbery kiss, but it's great because they're babies and that's why it's always cute, even though it's kind of gross. So it goes up and it licks its face, stays there for a second and looks to see if there's anything wrong. And if there looks like there's possibly anything wrong, it releases these perforants, which then will polymerize and form a hole in the embryo, in the enemies, in the cells, it's an enemy cell or abnormal cell. And then granzymes enter the cell. the enemy foreign weird cell. And the granzymes then degrade the cell's enzymes. And the cell dies by apoptosis. And then the cell that's dying, because the natural killer cell licked its face and then decided to kill it, is engulfed by a macrophage. So it goes up, licks its face, sits there for a minute, looks all over at it. If it looks like there's something odd about it, it releases a bunch of perforants, which makes little tunnels or holes into the cell that then allow granzymes through that destroy the cell's enzymes. And the cell dies by apoptosis. And then the dying cell that's dying by apoptosis is taken up by macrophages. Now, I'm sorry, I probably could have put this slide right here. I maybe could have did it twice, but I didn't. So I'm going to go back up here again. So bear with me here. And if we were going to talk about natural killer cells, we have to talk about how they kill. So coming back, inflammation. What's the point of inflammation? Why is it a good thing? It's a non-specific response to a tissue injury. And the ultimate goal is to bring in phagocytes and plasma proteins to the invaded or injured area of the body. And it isolates and destroys invaders, gets rid of debris, and prepares for subsequent healing and repair. I'm sorry I keep coming off on tangents, but I'll tell you about something really quick. So I learned when I was busy doing all this fostering with kittens and dealing with ringworm, the worst thing that you can do for ringworm is put an steroid on it or a cortisone on it. Why do you think that's the case? What would a cortisone do, or an anti-cortisol cream?
It's anti-inflammatory.
Say it again.
Isn't that anti-inflammatory?
It's an anti-inflammatory. So why is that so bad when you're dealing with ringworm?
Because like somebody said earlier, it probably like that kills all the bacteria then in that area.
Or it blocks this process from happening. When you're trying to get rid of something like ringworm, a fungal infection, and you block it with an anti-inflammatory, your body is intentionally trying to develop inflammation to kill the ringworm. And then you decide, well, you know what? It obviously is hurting, so we'll just stick inflammation. It hurts, so we'll stick some anti-inflammatory on it. And the reason I'm bringing this up is inflammation is a good thing. A lot of times it helps you to get better. I mean, it overall, almost always, most of the time, 99% of the time, it is helping. So inflammation, we get inflammatory chemicals like cytokines and chemotaxins, all being released. Cytokines, AKA chemotaxins, also known as chemotaxins. These inflammatory chemicals trigger mast cells. Mast cells release heparin. They activate signals from the blood, bringing in white blood cells. They work as a siren to bring in proliferating tissue cells that are in the tissue that are there to as part of the immune system. And they induce phagocytosis. Different signs of inflammation. This is a mnemonic I found online for signs of inflammation. Hyper. Heat. Induration of hardness, stiffness, toughness, or hardness of that area. Pain. Edema and redness. So a break in the skin introduces bacteria, which reproduce at the wound site. The activated macrophages that are already in that area engulf the pathogens and secrete cytokines and chemotaxins. These are those inflammatory chemicals. The activated mast cells release heparin. This little guy right here. Or not heparin, histamine. The histamine dilates the local blood vessels and widens the capillary pores. The blood vessel wall becomes sticky, causing the neutrophils and monocytes to attach. Chemotaxins that were released here by the macrophages attract neutrophils and monocytes, which squeeze out between the cells of the blood vessel wall, the endothelial cells of the blood vessel wall, in a process called diapetes, and migrate to the site of the infection. Yes. The monocytes enlarge into macrophages, and the newly arriving macrophages and neutrophils engulf the pathogens and destroy them. So a major part of inflammation is this diapetes of white blood cells coming in from the blood. So we get increased vascular permeability. And on top of that, vascular permeability, meaning things are leaking out of the blood vessels. One of the things that's coming out of the blood vessels are the leukocytes, the white blood cells, like monocytes and neutrophils, and basophils, and eosinophils, and whatever we need. And so the leukocytes, the way it starts working is we have what are called selectants that start being expressed on the endothelial vascular wall. And they bind to the leukocytes which have what are called integrins on them that are receptors that bind to the endothelial cells. And so the leukocytes get attached to the endothelial wall. They start to roll along the wall. They're just kind of diapetus, meaning they're moving. Putting out the pseudopodia, and they're moving along the wall of the vessel. They start rolling as they get better and better attached, and then they migrate through the vessel wall. Questions? So this is diabetes and how it's involved in inflammation. It's just essentially the leukocytes, how they are getting attracted to a site. Then they bind to these selectins that are on the endothelial cell wall of the vessel. And then the leukocytes start showing integrins on their cell surface, which helps with their attachment to the endothelial cell wall. And then the leukocytes migrate through the wall. Towards the chemotaxins. Think of the chemotaxins as being sirens, saying, this is where the infection is at, come here. So overall manifestations and outcomes of inflammation. Bacterial invasion results in the release of histamine by mast cells. The histamine causes local arteriolar vasodilation and local capillary permeability. We need that permeability so we get the leukocytes entering the tissues from the blood via that diabetes stuff that we just looked at. The local arteriolation, vasodilation causes increased blood delivery to that area. I'll tell you, this is why Vicks Vapor Rub is so good for acne, is because it increases the blood delivery to the tissue of the infected pore. So the increased local capillary permeability causes increased local accumulation of fluid, which results in increased plasmoid proteins out in the interstitial fluid, which includes the complement system proteins. And increased phagocytes in that area. The increased blood delivery also results in increased phagocytes because they're in the blood. Then there's an increased amount of phagocytic secretions, cytokines, that causes an increased defense against foreign invaders and ultimately tissue repair. And systemic responses such as fever. Any questions about inflammation? So tissue repair after the inflammation. Tissue repair can be perfect if cell division can replace the lost cells with the same kind of cells. This is where we get mitosis, and talked about mitosis in 121. This is where we get mitosis. One of the times we get mitosis. In non-regenerative tissue like nerve and muscle, Those lost cells are replaced with scar tissue because we can't regenerate with the same kind of cells. Drugs that suppress the inflammatory process, non-steroidal anti-inflammatory drugs like the NSAIDs, aspirin and ibuprofen, glucocorticoids suppress almost all aspects of the inflammatory process by reducing the production of cytokines. Think of cytokines as chemotaxins. And the glucocorticoids also reduce the body's ability to resist infection. I want to say also that they also resist your ability to detect cancers and destroy cancers for the same reason. Questions? We're essentially done with innate immunity here. Questions about it? I'm going to keep moving forward here. Just for a few more minutes here for the last 6 minutes of class. The two classes of adaptive or specific immunity, antibody-mediated or humoral immunity, involves the production of antibodies by B lymphocytes, also known as plasma cells. Cell-mediated immunity involves the production of activated T lymphocytes that directly attacks unwanted cells. So humoral immunity, or the antibody-mediated immunity, protects cells against extracellular pathogens and their toxins. Cell-mediated immunity protects the body against intracellular pathogens. So the humoral immunity detects, using B cells, detects intact antigens. And with the help of T helper cells from the cell-mediated immunity, results in plasma cells that secrete antibodies against the intact antigens, and memory B cells that will then be able to proliferate into plasma cells later if needed. T cells, rather than just free-living intact antigens, free-floating intact antigens, T cells go after antigens that are displayed by infected cells. And they defend against infected cells, cancers, and transplant tissues. So adaptive or specific immunity is mediated by the B and the T cells. It targets a particular pathogen. It's not always ready and it must be primed. It's systemic. It's got a stronger secondary response due to the memory cells. B cells come from lymphocytes that mature and differentiate in the bone marrow. T cells also come from lymphocytes that originate in the bone marrow, but instead of maturing in the bone marrow, they have to move to the thymus where they mature and differentiate. After early childhood, the new B and T cells are mainly produced primarily by B and T cell colonies established in peripheral lymphoid tissues. So B T cells start in the bone marrow. T cells differentiate and mature in the thymus. B cells differentiate and mature in the bone marrow. They then start to circulate in the blood and the lymph tissues. And they migrate around and proliferate throughout peripheral lymphoid tissues, like your lymph nodes. And upon activation, get stimulated. B cells are going to go on to perform the antibody-mediated immune response. And T cells are going to do the cell-mediated immune response. Questions? We've seen this slide before, so I'm going to just touch on it and then we're going to leave it alone. B cells, when we say they mature, they become immunocompetent. When T cells mature, they become immunocompetent. Most often, B cells and T cells get activated in the peripheral lymphoid tissues, like the lymph nodes, like the tonsils, like the spleen. And you guys, let's do this one. Actually, let's wait. We'll hold off on that and we'll do it next time. But plan on finishing the immune system next week. And come back to lab in an hour and a half because we're going to have fun today. I told you that I got some organs from a butcher shop. And so we're going to dissect the heart. And we're actually, if we have time, we're going to dissect two because I have a fixed one and a non-fixed one. And we're going to go through heart anatomy and talk about ECGs. So I'll see you in a little while, right?
Sounds good. Thank
Okay.
you.
Do you want us to email you our address for the urinalysis?
Email it to me or text it to me. It doesn't matter. And
okay.
I know that some of you have already done it, so I really appreciate it. I'll probably try to. It doesn't matter, Claire.
Okay.
Because probably what I'm going to do is I'll try to ask for them every week until I think I have everyone's.
Okay, I'll do it right now. I'm going to email you.
Awesome. All right.
Thanks so much.
You're welcome. I'll see you guys in a little bit.
Yep. All right. All right. Bye.
Bye.
So we're sending you our address for what again?
I am going to send you some urinalysis strips.
Okay.
Because I'm trying to make some of our labs as hands-on as possible, which isn't very easy in an online class.
Do you need a lot?
Toyin, just a sec. Just a sec. No, you're all right. Just a sec. So I'm trying to make our class as online as possible. Hands-on as possible. And a urinalysis lab is a good one where it can actually be kind of hands-on. But Toyin, what was your question?
Do you want us to send you what do you want us to send you?
Send me your mailing address.
Oh, okay.
I'm going to send you something in the mail. And once I've sent it out in the mail, I'll shred your address. I won't keep it around.
Do you mean to your cell phone?
Just your, I think Toya and I probably already have your cell phone number because I think you texted me, but I don't want that here. I just want your mailing address.
Okay, only our mailing address to your cell phone number, right?
That works, or you can email it to me. It doesn't matter.
Okay.
Whichever is easier.
Can you say your phone number? Because last time I tried to text you, it didn't go through.
Oh, really? Oh, it's 952 201 3126.
All right, thank you.
You're welcome. Zena, do you want to ask me the questions you had? Did you have questions?
Yeah, I was waiting for Katie and Safa if they had a question first, so I don't hold them.
Okay, let's see if they do. Katie, do you have a question? Alyssa? Give them a second. Because I know some people turn on their camera and then they walk away or they have internet issues or something. So give them a second.